Hello friends, Ace Button next episode. Now we are proceeding quick reference. And the chapter is Cell, the Unit of Life, under which 8.4.1, 8.4.2, that is cell envelope and its modification, ribosome and inclusion proteins. So mainly prokaryotic area. And remember, daily I am putting short question. One from first year, one from second year. Okay. So subscribe Ace Button, so you'll be getting all this. Already detailed notes are given in YouTube. Now we can proceed with the cell, the basic unit of life. For convenience, I will share the screen so it will be easy for you. I think screen came, so we can proceed. So quick reference. So we can start with the cell envelope and its modification, starting with the envelope. So envelope normally means envelope. That means a covering. Wherever it comes, it means a covering. And in most cases, prokaryotes have a covering. So I think idea clear what is envelope. Now cell envelope, we can proceed. Cell envelope, it has got two features. One, it is tightly bound. One, it is 3-layered structure. And 3 layers are written there, so it will be able to save time. Glycocalyx, cell wall, plasma membrane. Now, generally when we speak, we can find each layer has its own function. So each layer has to do its own function. Total together act as a protective layer. Now coming into classification, how bacteria has been classified. Mainly bacteria classified based on so famous classificatory mode based on stain and cell envelope. Stain and cell envelope. It is given by a famous method called gram staining by Danish botanist Christian Gram. According to that staining, 2 types, gram positive, gram negative. Gram positive means able to retain violet color that are written there to save time after treatment. While in gram negative, we can find after treatment, we lose the violet coloration. I think idea clear, so we can proceed to second page. Now, next is glycocalyx. Each layer we are starting. Glycocalyx differs in thickness. It's important. And composition in different bacteria. It can be 2 types. It is slime layer and capsule. Slime layer will be usually loosely arranged, while capsule is thick. It is very thick and tough. That is regarding outer layer. Now, inner to the outer layer, it is cell membrane. It is selectively permeable. So, after slime layer, capsule, then that is glycocalyx, then cell wall, cell membrane. This way it proceed. I written cell membrane first. It is selectively permeable. It actually interacts. With outside. Even though cell wall is there, cell membrane actually interacts with outside. Structure is similar. It is similar to. It is mean structure. Similar to eukaryote cell membrane. So generally glycocalyx, then cell wall, cell membrane. I written glycocalyx, then cell membrane, then only cell wall. So don't take in another manner. Outside is glycocalyx, then inside the glycocalyx, cell wall, then cell membrane. Now, outside the cell membrane, we can find cell wall. That is below glycocalyx. Shape of the cell. Determination. That is one function of cell wall. Shape of cell wall determination. It also provides mechanical strength. For what? Prevent bursting and collapsing of the cell. Bursting and collapsing of the cell. Okay, so glycocalyx, cell wall, cell membrane. Now next is mesosome. So the layers over glycocalyx, cell wall, cell membrane. I written as cell membrane, cell wall. So originally glycocalyx, cell wall, cell membrane. Okay, I think idea clear. Now mesosome. So when we come to mesosome, it is the extension of the cell membrane into the cell. Three forms. One is vesicles, tubules, and lamellae. And functions, it is connected with the formation of cell wall, formation, DNA replication. I think page is clear. Then after DNA duplication, its distribution helps in respiration, secretion, then increases surface area, increases the more action since surface areas increase. And in some, we can also find chromatophores seen having pigments. Now comes flagella. Flagella present under the condition if it is motile. Under motile condition only it is safe. That is flagella. It is cell wall extension. Extension. Three parts will be there. Which are the three parts? It is filament, hook. So filament, hook, and basal body. Basal body. Now comes pili and fimbriae. They are simply surface structures. Surface structures. That is pili and fimbriae. No role in motility. No role in motility. Pili elongated. Pili will be comparatively elongated with the protein pilin. With the protein pilins. Fimbriae, small, bristle-like. Bristle-like. And what is the function of fimbriae? Helps to attach. Helps to attach to rocks. To rocks and host tissue. Function is important. Directly NCERT. Now comes ribosome and inclusion bodies. See, ribosome and inclusion body connected with the plasma membrane. Specifically ribosome connected with the plasma membrane. The size of the ribosome will be 15 nanometer to 20 nanometer. 15 nanometer, 20 nanometer in size with the 50S and 30S subunit. So total 70S site for protein synthesis. Now single mRNA on which many ribosomes. Single mRNA on which many ribosomes. That forms what is called polyribosomes. Polyribosomes or polysomes we can find. I think idea clear. Now we can proceed to next page. Ribosome only this one. Now inclusion body. What is meant by inclusion body? It is a reserve material in prokaryote. It is reserve material in prokaryotes. Free in cytoplasm. Free in cytoplasm. No membrane. No membrane for it. No membrane. Now, typical example, phosphate granules, cyanophysin granules, glycogen granules. PCG. Now, gas vacuoles are seen in blue-green and purple and green photosynthetic bacteria. So, all reference made with NCERT. Quick reference only, details already there in YouTube, each area. Now, last episode regarding some values, I haven't not directly mentioned. Some I mentioned there, so for clarification, I put it here. That is regarding our third episode of quick reference. Cell envelopment and its modification, ribosomes and inclusion bodies. If you like this episode, subscribe or like it, share it, comment on it. We can meet later with the next part. For time being, we can wind up. Thank you. Take care. Bye.